Thank you, Søren and Alex. And now we will continue. Next is Anna-Maria from Enea from VP2 about the microbiome-based solutions in agriculture, please.
Okay, good morning. Now we are presenting the results obtained in Work Patch 2 on microbiome-based solution in agriculture. We start from the complexity of soil. Soil is a dynamic habitat, very complex, in which that hosts a large number of microbes, living organisms, that are essential for maintaining the biodiversity of the ecosystem above and below ground. And soil is important because from soil, food begins from soil, so it's fundamental for the crop production. Okay. And in SIMBA project, we aim to how we can use the biodiversity of soil to produce microbiome-based solution for agriculture. And as you can see from this graph pad produced by microbe support, microbes are everywhere in the food system, and in particular we are important in the soil, in plant, but also in the food. And they are responsible for the contribution to the transition towards the climate environmental friendly agriculture. So in SIMBA project, our question is how we can use this microbiome for agriculture. Microbiome is a complex name because it's composed by microbiota, the single microorganism, and the theater of activity. In other words, all proteins, all lipids, polysaccharides, nucleic acids, the environment they produced by microbes themselves. And so this interaction between microbes, their metabolites, and the environment is coded by microbiome name. And in SIMBA project, we focus on this attention, okay, how we can use these microbes from soil in order to increase the favor the sustainability in agriculture. Sustainability in agriculture can be used in two main approaches, by working on the management of soil and using new solutions by microbes that are present in the soil. In this way, we can favor the crop production, but also we can favor the healthy food and also the healthy people. And in SIMBA project, we started with the selection of microbial consortia that combined by beneficial microbes in order to favor the crop yield and crop quality. But we, at the beginning of the project, we discussed about how the procedure to obtain these efficient inoculants. And we started with the idea that if we combine different beneficial microbes with different functions, we are able to construct a syncombs that are combine different potential important functions for the soil plant productivity. And in SIMBA, we developed new microbial consortia with different important functions like nitrogen fixation, phosphorus solubilization, and biocontrol activity and plant growth promoting activity. And in this paper, we found the list of 3 important microbial consortia that were developed in SIMBA project, named MSCA, MSCB, MSCC, which are composed by one member of each important function like nitrogen fixation, biocontrol, and so on. And okay, we started with these 3 microbial consortia, and another microbial consortium was developed by the University of Copenhagen, named Microbial Consortium D. In particular, this consortium is able to favor the plant growth in drought conditions. And also in this case, we publish the results, so you are able for the entire community. The strains composing these microbial consortia are able to favor the plant growth in these conditions, and the effect is more respect to the single strains when used as single inoculants. And another activity that we perform in Work Patch 2 is important to define, okay, these strains were safe to be applied in the environments. In particular, we perform the whole genome sequence in order to exclude any risk related to the antibiotic-resistant genes or potential pathogenic virulence genes. And we demonstrate that our microbial consortia did not contain any determinant of virulence or antibiotic-resistant genes. The strains were, we performed the scaling up production in the ENEA laboratory in the south of Italy, and we performed the scaling up of the production by using bioreactor, and the strains in life-freeze condition were sent to the company, CCA in order to provide the inoculum for the field trials. We use different methods to deliver microbial consortia in greenhouse experiments and in field experiments. We start with seed coating or liquid suspension and moving towards char functionalization, power stabilization with zeolite. And in this slide, you can see the coated seeds that were used in field application in Italy and Germany. To reinforce the inoculum, we adopted also the use of arbuscular mycorrhiza fungi and bacterial compounds. So we composed the inoculum that was composed by different representative strains of arbuscular mycorrhiza fungi, also bacterial compounds that favor the plant growth, and in particular also favor the plant quality, the crop quality. We published the results of the positive effect of this consortia in greenhouse condition in heated and heated soil by University of Bono, and the crosstalk between plant and soil by University of Parma, in which we demonstrate that the plants respond to the application microbial consortia, favoring the increasing on targeted genes analyzed by transcriptomic analysis in leaves, in wheat and maize. So we demonstrate there is a crosstalk between microbes and plants, plant and soil. But when we move from the lab to greenhouse, and in particular to the field condition, we have to consider the abiotic stress that are present in field, and we tested this microbial consortia in 2 years in different field conditions and organic and conventional management in Italy and Germany. We found important effect on, in particular, Microbial Consortium C in field in 1 year, into 2020. Not so reproducibility effect in 2021, but anyway, when this consortium was applied in combination with mycorrhiza, we found a positive effect on plant growth. In Italy, the presence of bacterial compounds and char in different conditions, also in using the combination of bacterial compounds on tomato in the south of Italy, we found a positive effect in increasing on plant health in both conditions. In particular, in the experiments performed by University of Parma demonstrate that the microbial consortia favored also the increase on plant quality. So at the end, of this project, we found in particular the bioindicators from soil metagenomic studies. In particular, we revealed different taxonomic profiling from 16S RNA, but also from shotgun sequencing. We performed the metagenomic fragment recruitment, so we are able to determine GPM in soil when we applied in soil as inoculants. We are able to extract, as described before by colleagues from Guapage 1, the metagenomic assembly genomes from these metagenomics data, and also we are able to identify the important function of these microbes into the soil. So we identify the biomarkers of soil fertility. But also we identify the biomarkers of crop quality by investigating metabolomics from in fruit, so in particular in wheat and maize. We identify the metabolites that are strictly related to the crop quality. And in particular, we found a great nutritional quality when the plants were inoculated with microbial products. Okay, so the main products of Work Patch 2 is this SIMBA SYNCOM, so this synthetic community based by different microbial strains with beneficial microbes that are able to scaling up for commercialization. So the major innovation is the formulation of these promising microbial consortia, and also we are ready to register these microbial consortia for marketing. So in this slide you can see the main results obtained in Work Patch 2 from SIMBA SYNCOMs to the selection of different important delivery methods, to the evaluation of efficacy and reproducibility in field. We found the bioindicators, soil quality and fertility. In particular, we define a practical manual for stakeholders and farmers. The innovation exploitable results are present in this slide, and you can see that okay, we can focus the main attention on these microbial consortia, but these microbial consortia were able to send to commercialization and products production. And also, this microbial consortia was targeted as innovation radar from the European Commission. So we are ready to start with the commercialization and the production of these consortia for sustainable agriculture. Now I leave the floor to Daniel to follow the last part of this Work Patch 2.
Good morning to everybody. I would just like to shortly highlight one of the final outputs of the Work Package 2, which is a manual for testing the efficacy of PGPM applications. That's a deliverable 2.11. And just as a background, if you have a plant protection or crop protection agent that is admitted in the European Union, you will have to test it according to the EPO guidelines, the European Plant Protection Organization, and they give clear indications how the trials, the field trials, should be done. This also exists for microbial products which are crop protection agents, for example, Bacillus thuringiensis products, but it does not exist for what we call the new category of biostimulants. That's what we try to fill with this suggestion of having standards, and these standards I shortly summarize. So we have to carry out field trials on efficacy, and it's important to have the crop and site selection adequately. For example, if you are testing products which are supposed to increase the phosphorus availability, it's reasonable not to work on soils which have a high phosphorus supply already, because this will mask the effects. Another important point is the treatment selection and the mode of application, which need to be specified. And with respect to the treatment, it's especially important to have controls, such as positive controls, but also blanket controls, where you apply the inoculum without the additional material, but also try to have a treatment where you apply the additional material as well, just to see whether there is a nutritional effect. Because it's important that any type of biostimulants to which the PGPM belong don't have a direct nutritional effect by adding nitrogen, but by this biostimulating effect. Then important is the experimental design and the trial implementation, and afterwards something which in most publications you will see is only poorly described. That's a crop management. That means how were the crops treated? For example, did you apply herbicides or any other pesticides? And finally, the data collection. And for the data collection, we suggest to have data collection on the site. That concerns, of course, the soil conditions and the climate. But for the soil in particular, some additional information about soil humidity and soil aeration. And then anything related to crop growth, including nutrient uptake. And finally, crop yield and quality. You have seen in a figure earlier that there may be an impact coming on the quality of the product by changing the profile of the contents of the ingredients. And what would be advantages as well is to take rhizosphere samples to follow the fate of the inoculum or even changes in the soil microbiome. And finally, the data evaluation and presentation of the results is important as well to have clear indications. And just to finish, we have compared this to some recent publications, and we can see that some of the aspects that we suggest are already fulfilled. Are those lined in green here. But others are not, especially with respect to the control, which are often not as we would suggest to have also a positive control and a blanket control. And also if you look on the soil data, especially data on soil humidity and temperature, is often missing if you look on this data here. And so we are going to submit next week this, it's already submitted within the project, but it's also submitted to Frontiers in Plant Science, and we hope and we believe there's a high probability this to be accepted and to be usable for those who are working in field trials with this PGPM. Okay, thank you, and I think we have still now some opening points for the discussion.
Okay, and the last slide synthesizes the importance of microbial solution for the agriculture. So we put this slide in order to discuss and after this presentation. In particular, you can see in this slide we can use the microbiome as a predictive tools as soil fertility, soil quality, but also it's important also to consider the microbial products for the agriculture, to use the microbiome as a predictive agriculture, like for example in human we use precision medicine, and so we use precision agriculture by microbiome-based solution. And also we can use microbiome for the valorization of the agri-food system. So this is the advanced level, but below you can find how to deliver microbiome science and innovation. Important the investment in research, but also another important aim is the regulation of the microbiome products and how we can consider this tool integrating in one health concept from soil to plant to human. So I hope that this can stimulate the discussion later. Thank you very much.
Yes, thank you. I think that we have time for one question, if there is some.
Thank you for the talk. It was extremely interesting. I was wondering whether you have considered the viruses as a part of the microbiome and as a factor that can affect the efficiency of the probiotics as in the dairy industry and so. In this case, it will be bacteriophages, I mean.
Yes, okay. We can investigate this topic only when perform shotgun sequencing, because the viruses in the microbiome can affect the productivity. In our case, we did not have any virus, and also during the metagenomic analysis we did not find it. But anyway, the application of plant microbes can favor also the plant to resist to virus, to biotic stresses. So in our case, we did not find it because the plants grow without any virus application effect. So I don't know if Daniel can add an additional suggestion, but we did not consider, we did not found the virus in this complex microbial community. Is this the question? Yes.
Thank you. Just a quick question. When you were upscaling your syncoms, were you able to co-cultivate all of them, or how did you do it?
No, we cultivate single strains and compose the syncoms when we apply the Microbiome consortium into the field. So we decide to do it because if you decide otherwise to combine strains together, we can have also the competitive in vitro condition, even if in vitro conditions were tested before and the strains composed the Microbiome consortia were positive, so we did not show any competitive activity. But we decide to grow separately in order to have the strains at the same concentration before the application to the field.
Okay, thank you. Now, Fredrik, please.
Good morning, everyone. So I will be talking about work package 7. So I'm Fredrik Ang, and I will talk about the uptake of novel microbial solutions developed in SIMBA. So the focus of this talk will be on one central research question. So how can we, in fact, stimulate the uptake of microbial applications? So we have seen a lot of interesting, promising results on the effectiveness. So the next natural question is actually how we can take this up. So how can we make sure that the appropriate actors will take this up? Mainly, this will be a farmer's perspective, right? Because they are an essential actor in this food supply chain. However, it's also important to talk about the consumers, because what is being produced by farmers is going to be eaten at some point in time by the consumers. And we have a demand-driven supply chain. So the perspective is a bit different. So instead of focusing on technical issues, we focus here on an understanding from the perspective of social scientists, and in particular, well, economics and behavioral sciences. So let's dive immediately into results, interesting results, focusing on farmers. And this was a paper led by Annika Tensi, together with myself and Ine. And here we analyzed actually the drivers for adopting microbial applications in arable farms. So the application was in the Netherlands on the one hand and Germany on the other hand. We used what is called the behavior change wheel, the COM-B model, and we then analyzed also, well, what are the motivations and what are actually effective interventions to let this behavioral change wheel spin. What came out of it were 3 interventions. So first, it's important to create the right context, so the right norms and enablement. Secondly, what came out of it is that knowledge still plays a very important role, so education and learning are very important. And thirdly, last but not least, building trust is extremely important. And this trust can be built, well, by providing incentives. These incentives are not only economic incentives, such as subsidies, for instance, but also more behavioral steering. Now, the interesting thing is that the messenger played a very important role. So advisors play a very important role in this process to acquire up-to-date knowledge. In fact, what came out of it is a very big skepticism towards government, both in the Netherlands and Germany. So if you look at the farmers' networks, they trust their advisors, but not, well, more government-related instances. So it's very important to enable this dissemination and to provide clear advice through these channels. The second study that I would like to highlight is joint work with Annika again. She was a very good, diligent PhD student. And this focused on 98 Dutch arable farmers. And here we focused on the risk profile. So this is very important in economics. We look at, well, how risk-loving farmers are. So are they prone to taking risks or not? And here we see that, in fact, a lot of farmers are very risk-averse, and especially those risk-averse farmers are not really interested in applying these microbial applications. Another aspect of the study is that we had a test of a video that is being shown. It was an informational video. Many of you have seen this video as well. And this video provided information about microbial applications and the reasons why you should use it in farming. Well, so we had that one treated group, 50% were treated, and then 50% haven't seen this video. And what we clearly saw is that the willingness to adopt these microbial applications, well, are clearly related with seeing this video. Caveat is, of course, willingness to adopt does not mean that they will actually adopt it, of course. The study was conducted in the COVID-19 period, and we see that, well, that actually, well, a bit counter to our expectations, did not have an impact on the risk averseness of the sampled farmers. Interestingly, other studies have also confirmed this, so it did not lead to such a change in the risk profile. Now, Thomas Slijper, who is a postdoc, or was a, yeah, he's still a postdoc, but he moved to Sweden now. He led a paper which is published in the Journal of Clean Production, and he found also that knowledge is associated with sustainable agriculture practices more broadly. So knowledge is still very important here, like in our previous study. However, they also note that there is this result actually vanishes for practices that are already subsidized. So this may be important in the context of microbial applications as well. Now, so far we have talked about the production side. Of course, there are also consumers who have to consume. So we have actually also elicited the willingness to pay of consumers, almost 300 consumers from mainly Italy, Germany, and the Netherlands. And we focused on potato, wheat bread, and tomato sauce. The main finding is that there is, in fact, a positive attitude towards microbial applications in food products for the majority. Now, if you give the information how effective it is, especially in terms of substitution for synthetic chemical use, then you see that there is a willingness to pay for a premium. And this premium increases, so the willingness to pay increases, by, well, the effectiveness of these microbial applications. So if it can substitute more for these, for the synthetic chemical use, then consumers are more willing to pay. And also interesting was in the study that environmental concerns rather than true health concerns drive this result. So consumers do not seem to care that much about the health benefits of microbial applications. However, they are sensitive to more an environmental pitch of the product. So this can be also important in the marketing stage. Now, to conclude, am I very quick, or is it just me? No, it's okay. We see that we need, in fact, a careful targeting of farmers, which is required for the cost-effective stimulation of adopting microbial applications. So we see that the messenger, in fact, matters. So that's what we have seen in the first study. So if you want to encourage farmers to adopt microbial applications, at least for the study areas that we observed, so that was in Netherlands and Germany, we should focus on advisors rather than the government. However, I have to say that this is, of course, very context dependent. So I'm not entirely sure whether this would hold for Europe as a whole. So we should be careful and not over-interpreting these results. Now, again, this is maybe a regional result, but what we saw is, at least in the Netherlands, we saw that, well, relatively risk-loving farmers are more open to adoption. Okay, this makes sense, right? Because well, if you want to take risks, then you're also more open to adopt new innovations and to, well, be more a front runner from that perspective. And actually, well, I haven't stated that in the previous slides. We have also conducted a study in which we looked at, well, the relationship between like efficiency and innovativeness in general. So not only microbial applications, and what we see is that, contrary to our expectations, actually, there was a complete null result. There is no relationship between the innovativeness and the efficiency of farmers. So this makes it actually a little bit more difficult who we want to target, of course, because how are you going to find these risk-loving farmers? Well, it's not very clear how we can easily do this. So that is quite difficult. Now, from the promotion side, we do see that, well, providing more knowledge helps. And here I have to emphasize that this actually was interesting in the sense that this does not hold for all technologies in general. So here we see actually, and this is cross-validated apparently among, well, several studies that we have conducted, that, well, knowledge is actually a driver of using these microbial applications. And when you provide an informational video, this increases the knowledge. So, well, one might conduct that, well, this is actually in line with our reasoning that knowledge plays a role. So we do see that the willingness to adopt these microbial applications are actually very related to seeing this video. Now, from the consumer side, we see that, well, there is in fact a willingness to pay for consumers. Well, and, well, I have to say these studies, well, it's only one study. Actually, you should repeat these studies and adapt it to multiple contexts. But in fact, such a method can serve as the basis to actually set the prices for products that are treated with microbial applications more upstream in the chain. And, well, we used contingent valuation, which also has, well, some limitations in terms of that you can also have cheap talk, for instance, because people like to occur like morally good and will likely state a higher willingness to pay. However, if we look at it more carefully, we can actually segment these consumers. So we do see that environmental concerns are driving some willingness to pay. So this requires more investigation. And one can also validate this type of study with additional studies, such as if you conduct field experiments, for instance, then this hypothetical bias, as it is called, that you can have cheap talk, can be reduced substantially. Having said all of this, I think we should still be a bit cautious because so far, well, this talk was actually about how can we, well, make sure that the actors in supply chain will use these microbial applications. However, having seen all the other results, I would be a little bit careful in the sense that we still need substantial efficiency increases. So we need actually still some technological progress in terms of these microbial applications. As we have also conducted other studies within this work package, focusing more on the economics, so the costs of production seem to be very high compared to, well, the benefits that it yields. So there are some benefits, right? So you reduce your fertilizer use, you reduce your pesticide use, but okay, of course, you have increased costs because of these microbial applications as well. And so far, these investments, well, depending on the setting, you can have different results, so it might still be a little bit too high for economic viability. And especially, well, if we want, well, if there is more research, then we can also reduce this uncertainty about the purported benefits. So there are economic benefits and also purported environmental and health benefits. And if there is more clarity about these benefits, then it might also become clear to what extent you can increase the sustainability of agriculture, mainly by these microbial applications. However, we did find some limited evidence in other work packages on the reduction of greenhouse gas emissions and pesticide use and the increase in yield. So that is a good silver lining. So that is, I think, a more optimistic sign. But again, we need more research and development to make sure that these microbial applications are very effective also in all sustainability terms. Thanks.
Okay, thank you, Fredrik. Now we have time to questions.
Thank you so much for this really interesting presentation. I think really going to the end user and stakeholder is crucial. I was just wondering on the examples that you brought, like Netherlands and Germany, who are the advisors? Who is advising farmers in regard of the microbiome applications?
Yeah, well, interestingly, in the Netherlands, it used to be actually governmentally regulated, but then it privatized at some point in time, I think in the 80s. So these are private organizations, and I think there are some mechanisms that you can get, some subsidies to get advice, but by itself, it's actually privatized. So private companies are actually doing this.
Because I think keeping independence in this sector and not being like one, because I know in many countries, farmers' advisors are actually representatives of different companies who then advise towards their product. And I think it's going to be crucial to keep this balance, not to get them falsely advised. Yeah.
Exactly. So I would very much be very cautious to look really at the context. For instance, in Ireland, you have a very different setting, even though there is, you have the same things going on, cows running around, like in the Netherlands. But the advisory services are very different. So it's really, from my understanding, it's really the government that steers this extension, which is, different. But it's a good point. Thanks.